Welcome, everyone. I'm Carolyn Drummond with the Healthcare Partnerships team at MDA. Thank you for joining us today for the MDA Industry Update webinar presented by Sarepta Therapeutics. And thank you for all you do for MDA and the neuromuscular community. MDA's Clinical Industry Update webinars are programs which provide opportunities for the medical community to receive information and updates impacting care and treatment directly from pharmaceutical, biotechnology, and medical equipment companies. These webinars are just a part of MDA's medical education resources. We encourage you to visit the medical education pages of MDA.org. You will find resources such as considerations for care, case studies, recorded grand round webinars, research grants, and so much more. The intention today is to discuss pre-existing antibodies prior to gene therapy. Sarepta will not be discussing details on any approved or investigational therapies. Now it's my pleasure to turn it over to Sarepta Therapeutics for today's webinar.
Great, thank you very much, Dr. This is Teji Singh from Sarepta Therapeutics. I'd like to thank MTA for hosting this webinar and to all of you for joining. Happy afternoon or good morning, depending on the time zone that you are. I'm here to discuss the pre-existing antibody testing prior to AAV gene therapy. Before I get into that, I would like to just discuss a little bit about gene therapies themselves. I just had, one technical thing to advance the slide. So basically, there's three essential components for gene transfer therapies. That's the vector, the promoter, and the transgene. The vector is the delivery system of getting the promoter and the transgene to the target cell. The vector itself drives safety in biodistribution. The choice of vector is dictated by what disease you want to target in terms of what cells you want to deliver to. Within the vector is the business end of the gene therapy. That's the transgene. The transgene is responsible for actually providing the coding sequence for the protein of interest. That protein is what leads to the function of that gene therapy. So it's an important determinant of the functional impact of the therapy itself. The promoter is something that actually turns on that transgene, and it's very selective. It can turn on transgenes in specific cell types, or it can actually less express them in other non-target cell types. So it drives the expression. When we're talking about pre-existing antibodies, we're really focusing on the antibodies to the vector. Now, what type of vectors are there? Multiple types, but AAV vectors, or adeno-associated vectors, are the most common ones in clinical research and also now in commercial use. The neat thing about an AAV vector, it is a non-functional virus. The wild-type AAV has within its genome or sequence replicant and capsid proteins. So it has the ability, in the presence of another virus, to actually replicate and produce new capsids. So it can produce new versions of itself. In the vector, those replicant and capsid proteins, or those sequences, are removed. So the AAV vector does not have the ability to actually self-replicate, and it cannot express these wild-type replication proteins. That makes the AAV vector a unique way of actually administering a gene therapy to a cell without worries about replication. We've used AAVs for over 30 years. The reason that AAVs are used is they're generally non-pathogenic viruses, don't really cause human diseases. There's multiple types or serotypes that allow you to actually target, because of their trophism, specific types of tissues, cells, or organs. So you can actually tailor the vector that you pick for the disease state that you want to treat. They have relatively low pre-existing immunogenicity. I'm going to use a little air quote for there because we're going to talk about the differences in immunogenicity by capsid, but overall, compared to other delivery systems, they're not very immunogenic. And they can be used to deliver to replicating and non-replicating cells. So as I mentioned, they're not strongly immunogenic, but they do elicit a cellular and humoral immune response. This is still a foreign protein, so the body is geared over time to react to foreign proteins. So you will have an immune response. That's why for a lot of gene therapies, they're usually administered in conjunction with an immunosuppressant, typically steroids, just before the infusion and for a period of time after, determined by what vector you're using for that particular disease. Now, pre-existing antibodies refer to those that exist before an exposure to a vector, so before actually an infusion. After infusion, everybody has antibodies, but pre-existing are those that exist naturally to a specific type of vector. And the type of antibodies that exist can be neutralizing or they can be total binding, so any sort of antibody, which I'll go into more detail later. In the case of Sarepta's product, we use the AAV RH74 vector. So when we do screening, we're screening for pre-existing total antibodies to AAV RH74. This slide just demonstrates the differences that exist for the types of vectors. They're basically to highlight the fact that there's more than one type, that these types that you use for a particular disease are based on their tropism, their affinity for certain cells, tissues, or organs, and that determines, what disease that they would be used to treat. On the far left, you see the AAV RH74 platform, which Sarepta is using for DMD and limb-girdle. We've chosen that because it has a high tropism for skeletal muscle, respiratory muscle, and cardiac muscle, which is the muscles that are involved in DMD. On the far right, you see the AAV9, which is isolated from humans, also used in DMD, but this vector can also target other neuromuscular diseases because it can cross into the CNS system. What does the RH74 stand for in terms of the Sarepta vector? RH is because it's isolated from rhesus monkeys. As you can see here, AAV74 and AAV8 both come from that source. The others come from human sources, and that can actually impact to some degree the pre-existing immunity that exists. Like I was mentioning, the screening is done for pre-existing antibodies. There are two types of screening. There's the total antibody assay, which detects all total binding antibodies. So these include neutralizing and non-neutralizing. They detect any antibody that binds to the AAV vector. Doesn't differentiate between the two types. Because of the way this test is done, which I'll explain shortly, the turnaround times for results are a lot shorter. Neutralizing antibodies look for transduction interference. They measure the ability of that vector being able to get into a cell system. So they actually measure, when we say neutralizing, that antibody prevents that transduction, also prevents that efficiency. These results are influenced by the cell line that you use, because some cells actually transduce easier than others, and because there's more manipulation and steps involved, they take a longer turnaround time. And because it's hard to automatize some of those steps, the amount of throughput, how many tests a lab can get through, is also lesser with a neutralizing antibody. What is the consequences of an antibody response or a pre-existing antibody? Well, neutralizing, very simple. That blocks transduction. It binds the capsid in such a way that it can't bind to an AAV receptor on the cell surface, preventing that capsid from actually getting into the cell and unloading its package, its promoter, and its transgene. But both neutralizing and total binding antibodies can act as opsins, basically a marker on a capsid surface that leads to opsonization, which means basically makes that capsid more prone or more marked or identified for phagocytosis from macrophages, etc., or indirectly by stimulating complement or cytokines or inflammatory cytokines that will also bind to the capsid, making it more identifiable for phagocytosis or lysis. Both also can directly or indirectly lead to complement activation. The activation indirectly would be when the cell or the capsid is destroyed, it can release split fragments that lead to the complement system being activated, or the antibody can actually attach to another complement driver that leads to a complement response. The amount of response or the types of response determines the side effects or the clinical findings that you would see with an infusion after an existing antibody is identified. Going to the ELISA technique, I mentioned previously that this is an assay that looks for the binding of all antibodies to an AAV, both neutralizing and non-neutralizing. It's a highly sensitive and simple assay. ELISA stands for enzyme-linked immunosorbent assay. So there's an antibody to the antibody that's linked to an enzyme that does a chemical reaction that either leads to a fluorescence, an immunescence, or a colorization or a colorimetric response. So this is something that a machine can read the response to. So this is something that you can automate. So you can do many, many samples, run them all concurrently. You can also have rapid turnaround. This is a highly sensitive test. And the serodilutions can be used to actually quantify the total burden of antibodies. So then this makes this, the properties of this makes it a very, very suitable test for diagnostic use. The ELISA method, basically, the way it works is that you take your sample, you put it into one of the wells, usually it's a 96 well plate, and then the initial sample is diluted by 100% or diluted by half. So if the initial sample is 1 in 25, it's now 1 in 50. Then you do a series of dilutions, 1 in 100, 1 in 200. The cutoffs can vary by where you start from, but basically you're doubling each of the dilutions. And what you determine as a result is when you actually have the titer or the cutoff where you saw a response, a chemical response, or in our case, a fluorescent response, and when you don't see a response. So the last dilution that produces a signal and the first dilution that ceases to produce a signal determines the titer. Now, with any given program, the cutoff of what's considered seronegative or seropositive is a titer value that you established in your research program, your non-clinical and clinical program that had some sequelae, either lesser transduction or an adverse event profile that was not favorable. And that is your cutoff for what you would consider a negative or a positive result. In the example of RA74, that cutoff is 1 in 400. And if you look at sample 1, or the sample from the first subject, you can see that there was multiple dilutions needed before you actually don't see a signal, which puts the titer at 1 in 1600. This is beyond the cutoff, so that test would be recorded as seropositive. On sample 5, there's 3 dilutions, with the fourth one being now negative. That puts the cutoff at 1 in 100, or the titer at 1 in 100, which is below the cutoff, so that would be considered seronegative. Individual assays will have different cutoffs determined by what was found in their preclinical and clinical program. So 1 in 400 for RA74 is not the same as a titer for an AAV9 or an AAV8. It would be determined specifically by the assay for that particular program. That is for an ELISA to look for total binding antibodies. What we normally do for neutralizing antibodies is you do a transduction inhibition assay. Now this actually measures the ability of an AAV vector carrying a transgene to transduce cells in an in vitro or in vivo system. Typically in vitro in the commercial setting where you're doing this in a test tube, and an in vivo would be using an animal model. In this system, what you do is you take the AAV vector or the capsid of interest, you put into it a reporter gene, a gene that actually leads to some signal being produced in the cell when the cell is transduced, and that's a signal that you can detect. So luferase detects basically a chemical reaction that shows up as a color. You take that vector and you incubate it with the patient's plasma. If the patient's plasma contains neutralizing antibodies, it will bind to the vector and prevent it from infecting the cells in that test system, that in vitro system. And if it prevents it from getting into the cell, you don't get the readout of that reporter gene. That gene can't actually produce the reporter protein, so you don't see it. So that would be a positive response to show that there is an AAV neutralizing antibody to that specific capsid. If it does get in, you will get a signal, and that signal will say, well, there isn't one, or if the signal is weak, that is actually not fully neutralizing. As you can tell, there's multiple steps here compared to the ELISA technique, so this is actually more labor intensive, requires some manipulation, and because it's a cell system, it's not as efficient as a mechanical system that's just with a chemical reaction. So these tests take longer to report and are also less sensitive. Just going to defer it a little bit now for the timing of when testing should be done. What I want to actually point out right here is that initially at the newborn to up to about 3 months of age specifically, but as long as up to 6 to 12 months, there's actually 2 antibodies that exist in the newborn. There's the newborn's own antibodies that are developing. The immune system is, starting to actually gear up around 2 to 3 months of age. But there's also passive placental transfer of maternal antibodies. So in the initial phase, there may be positive results, but these are driven by the presence of maternal antibodies. So you might have to do additional testing to separate out the 2. The other thing is for AAVs, because they exist in nature, there is a chance that over time you have a longer duration of environmental exposure. So prevalence for certain AAVs, not all, will actually increase with age. So by a certain age, you've been exposed for such a long time that your total prevalence to a neutralizing antibody or a total antibody is quite high because you've had that duration of time for that natural exposure to occur. Going back to a theme that I discussed previously, you can tell that there's different methods of actually testing. There's different titers which each program establishes. So the entry into any specific gene therapy program requires a unique sponsored endorsed antibody test. This is something that the sponsor has worked on in the clinic, and then that's validated by a series of sort of validation steps to show that this is actually the assay that should be used for that program. So one program, one test sort of paradigm. Now, the eligibility requirements for a program have multiple factors, but in terms of the assay, they will have different cutoffs. That could be based by the different techniques or the reagents used in that particular assay, the methodology, total binding versus neutralizing, how the assay was validated against the various sort of test samples it was used. It's going to be determined by the serotype. Different serotypes or AAVs will have different eligibility requirements. And of course, the detection methods. Like for example, in ELISA, there's 3 different detection methods. So it could be an ELISA method, but it may be using one of the 3 different methods. So it'll be unique to the methods that's used. In Sarepta's case, we have the ELISA method looking for Rh74 antibodies. And we did a study, a phase 2 study, to one, validate the assay that we're using commercially, or will be using commercially, but also this study helped us understand the existing seroprevalence or the existing number of patients with pre-existing neutralizing antibody, total binding antibodies. So this was a study of 101 boys that we did, that were 4 years or up to 18. These boys had a single blood draw done, either at home or at a local lab. And then we basically analyzed the blood for the presence of antibodies, with an endpoint being the percentage of subjects, percentage of patients, with a titer greater than or equal to 1 in 400. So the results of this study showed that in the US, this was a US-based study, there was an 86.1% of the samples of the boys were seronegative. So a high percentage, 86.1%, would have been less than 1 in 400 in terms of the results that they had. So that puts us as a prevalence pretty much on the better end compared to certain ones in this age range. But that's not, again, very much driven by the various types of capsids. Different capsids will have different seroprevalences. Sarepta, based on these results, quantified this commercial assay. This is now an assay that we'll be using going forward for our commercial program. And we're actually offering the DMD community a sponsored testing program to measure for pre-existing total binding assay antibodies to AAV Rh74. This is for subjects that actually complete an enrollment form and qualify for the treatment. It can be initiated through us, through the Sarepta Assist program, or through the diagnostic partner, Athena Labs or Quest. And this would result in the results being available to the families within about 5 to 6 days after the test if they qualify via the enrollment form. So in summary, pre-existing antibody testing has several criteria. It can impact gene therapy eligibility. It's not the only basis for eligibility. There's other sort of parameters that will be depending on the program for what leads to eligibility for that particular product. All patients should be tested for pre-existing antibodies prior to AAV-based gene therapy because of concerns about both transduction efficiency and safety. Only a manufacturer-endorsed antibody test can be used to determine eligibility for a specific gene therapy program, and that's because of all the steps it takes to make sure that that test is specific for that indication, that program, that capsid, that vector. And as I mentioned, Sarepta is offering a sponsored testing program to detect pre-existing total antibodies to the AAV Rh74 vector for our commercial products as they become available. With that, I'm going to say thank you and open it up to questions. They're all via the chat, so you can enter it in the chats place, and I'll defer to Gina or Caroline or Luca to read them out if any exist.
Yes. If you have questions, please just enter them in the chat. We'll give it just a second, and then we'll continue on. So I'm not seeing any questions come in. What great information shared today. Thank you, Dr. Singh and Sarepta, for the insightful presentation, and to our healthcare professionals for attending today's program. We hope that you will join us for future industry update webinars. Have a great day, and thank you.
All right, thank you. Bye, everyone.